I want you to just imagine an organism for a second. An organism that is, for all practical purposes, immortal.
Oh, wow. Okay.
Yeah, think
Yeah.
about something that can actually survive boiling water. Something that, just laughs off radiation. You can hit it with standard chemical disinfectants and it does absolutely nothing.
Right. It just ignores it.
Exactly. It just
Yeah.
sits there buried in ordinary dirt, waiting patiently for decades. I mean, sometimes longer than a human lifetime, just waiting for the absolute perfect moment to strike.
I mean, honestly, it sounds like a monster from a science fiction movie.
It really does.
But it is very real. And there's a pretty decent chance it's sitting in the soil right under our feet today.
Right. So today's deep dive is all about Bacillus anthracis, which you probably know better as the Anthrax Bacterium. And we are pulling from a really massive stack of sources today to try and unpack this thing.
Yeah, it's a lot of material.
It is. We've got 19th century medical diaries, we've got declassified FBI investigative reports, and some incredibly dense modern genomic research papers.
Right, which we will try to make sense of.
We will definitely try. Our mission here is to figure out how this microscopic organism essentially birthed the entire field of modern medical microbiology. We're going to look at the genuinely ingenious biological weapons it carries, debunk some of those persistent Hollywood misconceptions about how you catch it, and, well, figure out why it remains a top-tier modern biodefense threat.
Setting the stage here is just crucial for anyone listening.
Yeah.
Because to understand infectious disease today, to understand how doctors diagnose you, how they treat you, how we even conceptualize illness itself, you really have to understand the legacy of this specific bacterium.
Yeah.
It is the absolute bedrock of modern medicine.
And before we even get into the mechanics of how this bacterium kills, which, by the way, is a wild story on its own, the discovery of its mere existence completely rewired how humans understand sickness.
Right.
Because anthrax isn't just a disease, it's literally the foundation of the germ theory of disease.
Yeah, if we look at the bigger picture of the 19th century, medicine was, well, largely operating in the dark. Doctors believed in miasma.
Ah, the bad air theory.
Exactly. The idea that bad air made you sick. But then, around 1850, a French physician named Cassier de Vane and a Swiss researcher, Pierre Rayer, they started looking at the blood of farm animals.
Because these animals were dying of what the French called a charbon, right?
Right, or what English farmers called splenic fever. And under their primitive microscopes, de Vane and Rayer noticed these strange, rod-shaped bodies floating in the blood.
But I mean, just seeing something at the scene of the crime doesn't prove it committed the crime, Like, you could find a bystander at a bank robbery, but that doesn't make them the bank robber.
That was the exact problem they faced. They observed these rods, but they couldn't definitively prove those rods were the killers.
Right.
Later on in the 1860s, de Vane actually managed to show that blood containing these rods could transmit the disease to healthy animals.
Okay, so a step closer.
The step closer, yeah. But it was still an incomplete demonstration because the biological link was missing. No one could prove the rod itself was alive and, actively causing the disruption.
So here's where the narrative gets just completely fascinating to me. Enter Robert Koch. It's 1876.
Right.
He's not at some grand, well-funded university in Paris or London. He is literally a country doctor in Prussia, working basically as an amateur scientist in a makeshift home lab.
And Koch did what no one else had figured out how to do. He isolated the bacterium completely away from any animal.
Wow.
Yeah, he grew it through multiple generations in pure culture, just sitting on a glass slide. And his setup was, well, brilliantly resourceful.
Right, the feed-in part.
Yeah. To feed the bacteria, he used the aqueous humor from an ox's eye as his growth medium.
Okay, wait, I have to pause here. Aqueous humor. Are you saying he essentially squeezed the fluid out of a cow's eyeball to feed his bacteria?
That is exactly what he did.
Oh my God.
I know. It is the clear fluid that fills the space in the front of the eyeball. It's nutrient-rich, and most importantly, it's sterile. Koch realized it was the perfect liquid to keep his microbes alive outside of a body.
That is intensely MacGyver of him. So he grows the bacteria in this eyeball fluid. He watches it form these dormant spores under his microscope, and then he stores those spores for months,
Right.
far away from any animal, far away from any fresh blood. And then he takes those stored spores, injects them into healthy mice, and suddenly the mice come down with fresh anthrax infections.
Yes. And by doing that, Koch answered the most important medical question of his era. How do we definitively prove a microbe causes a disease? This experiment is what gave us Koch's postulates.
So Koch was like a detective who didn't just find the murder weapon at the scene, but practically recreated the exact crime in a lab to prove how the weapon was used.
That's a great way to put it. His criteria became legendary. Find the organism in all cases of the disease, isolate it in pure culture, cause the disease by introducing it to a healthy host, and then re-isolate it from that new host.
So thorough.
It is. That sequence is still the gold standard taught in medical schools today. It was so compelling that Koch presented this to a famous botanist, Ferdinand Kohn, and Koch's career just exploded overnight.
Right. He went on to identify tuberculosis and cholera.
Exactly, eventually winning a Nobel Prize.
Wow. But so Koch figures out the cause, but the story doesn't end there. 5 years later, Louis Pasteur steps back into the spotlight. It's 1881, and Pasteur decides to do this massive public science demonstration at a farm in Pouilly-le-Fort, France.
Oh, this event was pure scientific theater. Pasteur wanted to test if a heat-attenuated, meaning, a deliberately weakened strain of the Anthrax bacterium could work as a vaccine.
I just try to picture myself as one of the skeptical farmers or journalists in that crowd, because Pasteur takes 24 sheep, a goat, and some cattle. He vaccinates them with his weakened strain,
Right.
and he leaves a matching group of animals completely unvaccinated. Then he takes a syringe full of a fully lethal, virulent strain of anthrax and injects every single animal.
Yeah, it was a massive gamble.
And he just tells the crowd, like, come back on June 2nd.
The tension over those few days would have been absolutely unbelievable. The medical establishment was basically ready to laugh him out of the profession.
So what did the crowd see when they actually showed up on June 2nd?
They arrived to find an absolute miracle of modern science. Every single unvaccinated animal was either dead or visibly dying in the field.
Wow.
And every vaccinated animal was perfectly healthy, just grazing normally. It proved that germ theory wasn't just some academic descriptive concept for guys with microscopes.
It could actually be harnessed to protect you.
Exactly. It proved the principle of vaccination could be a practical, generalizable medical technology, scaling way beyond what Edward Jenner had done earlier with smallpox.
The thing is, the fact that Koch and Pasteur could even study and manipulate this organism so predictably, it hinges entirely on its rugged physical properties. The biology of this bacterium is just baffling to me. It is built like a microscopic tank.
It really is. And the core of that durability is what we call the endospore. The spore is essentially the organism's ultimate survival mechanism.
Break that down for someone who hasn't, taken a biology class in a while. What actually happens when this bacterium is just out in the world and its environment gets hostile?
Well, imagine your normal vegetative cell of Bacillus anthracis. You are swimming around, multiplying, and suddenly you run out of nutrients, or maybe the temperature spikes or everything dries up.
Right.
Most bacteria simply die in that situation. But this organism initiates an incredible transformation. It dehydrates its own core, it shuts down all its metabolism, and wraps itself in multiple protective protein coats.
Just totally shields itself.
Yep. And on top of that, it builds this tough cortex made of peptidoglycan.
Peptidoglycan, what is that actually doing for the cell?
Think of peptidoglycan as a highly rigid, mesh-like armor made of interwoven sugars and amino acids. It reinforces the spore so it can withstand immense physical stress.
Okay, so it puts itself into a heavily armored cryogenic sleep.
Basically, yeah.
And in that inert state, it can survive direct sunlight, it can survive boiling water, and it can sit in ordinary soil for decades. We actually know this for a fact because of Grunard Island, which is a detail from our sources that completely blew my mind.
Oh, the Grunard Island case is a very dark chapter of World War II research.
Yeah.
In 1942, British military scientists were developing anthrax bombs. They tested them off the coast of Scotland, deliberately contaminating this small island with spores.
And the aftermath of that test really proves how terrifyingly durable that peptidoglycan armor is. Because the island remained absolutely deadly and entirely off limits to humans for 48 years.
Yeah, almost half a century.
It took until 1990 to finally clean it up. And the decontamination effort was massive. They couldn't just spray it down with a hose. They had to soak the entire topsoil of the island with 280 tons of formaldehyde solution diluted in seawater just to declare it safe enough for the original owners to return.
It's wild. In some documented outbreak zones around the world, soil-based anthrax spores are estimated to remain viable for many, many decades.
Wow.
Theoretically, they could last a century or more under the right conditions. But here's the thing. The spore is only the armor. The real threat lies in the genetic weapons it carries inside.
Okay, and this is where I struggled a bit with the reading, so I want to make sure I really understand this. The lethal traits of anthrax, the things that actually kill you, aren't even on its main chromosome.
Right.
They live on these two extra chromosomal rings of DNA called plasmids, specifically plasmids PX01 and PX02. It's almost as if a totally harmless bacterium found a couple of genetic USB flash drives lying around and just downloaded a highly advanced two-part weapon system.
That analogy is biologically spot on.
Really?
Yes. The bacterium uses these two plasmid systems in tandem to do its damage.
But wait, if the deadliest parts are in its main DNA, what happens if the bacterium loses those USB drives? Like, what is it without them?
It becomes nearly harmless. Yeah, if you strip a Bacillus anthracis cell of PX01 and PX02, it loses its ability to cause the severe disease we recognize as anthrax. Losing just one of them dramatically reduces how dangerous it is. And veterinary vaccines today actually rely on this exact concept.
Oh, like the Stern strain that was mentioned in the veterinary literature? I saw that come up a few times.
Yes, exactly. The Stern strain, which is used to vaccinate livestock, lacks the PX02 plasmid.
Okay.
That means it can't build its protective camouflage, but it keeps the PX01 plasmid so it can produce just enough toxin to train the animal's immune system without actually killing the animal.
Okay, let's look at those two genetic USB drives then. Let's start with PX02. What does that camouflage actually look like?
So plasmid PX02 codes for a structural capsule that completely surrounds the cell. Now, most bacterial capsules are made of sugar-based polysaccharides, which immune systems are pretty good at recognizing.
Right, scientific stuff.
But the anthrax capsule is made of poly-D-glutamic acid.
Which is an unusual, protein-like polymer, right?
Very unusual. And because of this unique protein-like structure, the host's immune system is basically blind to it.
Wow.
The phagocytic cells, the immune cells roaming your bloodstream, whose entire job is to eat and destroy invaders, they simply don't recognize the anthrax bacteria as a threat. They bump into it and just ignore it.
So the bacteria can just multiply completely unchecked.
Exactly.
It's essentially wearing an invisibility cloak. And then you have PX01, which is the actual payload, the anthrax toxin itself. And from what I read, this toxin isn't just a single poison. It's incredibly sophisticated.
Right.
It's 3 separate proteins that are harmless if you encounter them alone, but devastating when they link up.
Yeah, the 3 proteins work like a coordinated strike team. First, you have protective antigen, or PA.
So PA is like the battering ram that breaks open your cells.
It is actually more like a Trojan horse. PA binds to receptors on the outside of the host cells and essentially forms a microscopic pore, basically a tunnel through the cell membrane. Its only job is to act as a ferry to push the other 2 components inside.
Okay, so once that microscopic door is propped open, the other 2 rush in. What are they doing once they actually breach the cell?
Well, the second protein is lethal factor, or LF. When LF links with PA, it forms what we call lethal toxin.
That doesn't sound good.
It's not. Lethal factor is a protease, which means it's an enzyme that slices up other proteins. It specifically targets and cripples the host's immune signaling pathways.
Oh.
And in severe cases, it triggers sudden, catastrophic cell death. Your cells basically self-destruct.
And the third one?
That's edema factor, or EF. When EF combines with the PA ferry, it forms edema toxin. This enzyme completely disrupts how your cells regulate water.
Oh, wow.
Yeah, it causes massive fluid swelling, or edema, which is a hallmark of severe anthrax infections.
Okay, so just to recap, we have an invisible, immortal tank that sneaks into your cells, shuts down your internal alarms, and causes your cells to either spontaneously die or swell with massive amounts of fluid.
That sums it up.
Which naturally leads to the question, how does an average person actually encounter this thing? And I think the biggest misconception we need to clear up right away is the Hollywood version of anthrax.
Oh, absolutely.
Because it does not spread person to person like a cold or the flu.
That is a critical point. You cannot catch anthrax through casual contact or coughing or sneezing from an infected person.
So if you are, say, sitting next to someone on a bus who happens to have anthrax, you are entirely safe. There is no infectious cloud radiating off of them.
Right.
You have to have direct exposure to the spores themselves.
Right. The spores are like microscopic dormant landmines scattered in the soil. They don't actively hunt you down. You have to accidentally step on them or dig them up or breathe them in.
Yeah.
And the reality is humans aren't even the primary targets for these landmines. Anthrax is fundamentally a zoonotic disease. Its natural cycle belongs to grazing herbivores, cattle, sheep, goats. They ingest the spores from the soil while they're just out eating grass.
So when humans get it, we are just incidental, quote-unquote, dead-end hosts. We just stumbled into the agricultural crossfire.
Exactly.
And the way you stumble into that crossfire determines everything about your chances of survival. Let's trace how a farmer might actually get this. The most common way, historically at least, is getting it on your skin. Cutaneous anthrax.
Right. Cutaneous exposure accounts for about 95% of naturally occurring human cases. The spores enter through a small cut, a scrape, or, a break in the skin while handling infected animals or their hides.
And the resulting infection is actually what gives the disease its name, right?
Yes.
Because it causes this painless black necrotic ulcer on the skin called an eschar. And the Greek root word anthrax literally translates to coal, referring to that distinct coal-black lesion.
That's right.
But I mean, as ugly as it looks, if you get treated with antibiotics, cutaneous anthrax is highly survivable. The fatality rate is under 1%.
It is. But things get much, much worse if the exposure happens internally. Gastrointestinal anthrax occurs when someone eats undercooked meat from a contaminated animal.
Oh, man.
It is vastly more dangerous because the bacteria are multiplying inside the really rich environment of the digestive tract.
And then there's the truly terrifying one, inhalational anthrax. Breathing the spores deeply into your lungs. Historically, without early and aggressive antibiotic treatment, the mortality rate for inhalational anthrax was around 85 to 90%.
Yeah, and this form had a very specific occupational name in the 19th century. They called it wool sorter's disease.
Wool sorter's disease.
Right. Imagine you are an English textile mill worker in the 1800s. You spend 12 hours a day in a poorly ventilated room sorting imported animal hides and wool. Those animal products were often heavily contaminated with dirt and dried blood containing spores.
So by just shaking out the wool, they were unwittingly aerosolizing millions of these invisible landmines into the air they were breathing.
Yes.
Even today, despite having modern intensive care units and antitoxin therapies, inhalational anthrax requires immediate, massive intervention to save the patient.
It really does.
And strangely, our sources highlight a fourth route that is incredibly rare, but it really shows how opportunistic this bacterium is: injection.
Yeah, this is a relatively new clinical form, primarily identified in a cluster of heroin users in the UK and Germany around 2009 and 2010.
Right.
The prevailing theory is that batches of the drug were contaminated with spores, perhaps from bone meal used as a cutting agent, or maybe it was contaminated during transport. The users were bypassing the skin and the lungs, injecting the spores directly into their soft tissue.
And because these microscopic landmines are so devastating when they get into the body, and because they are so stable to store in a warehouse, humans inevitably try to weaponize them. We took a natural agricultural hazard and engineered it into a modern security nightmare.
We did. And that transition from a natural disease to a state-sponsored bioweapon brings us to some of the most sobering case studies in modern public health.
I really want to spend some time on the Sverdlovsk incident in April 1979, because when I read the declassified reports on this, it read like a thriller, but it's really this tragic textbook cautionary tale.
Sverdlovsk, which is now the city of Yekaterinburg in Russia, was home to a massive, secret Soviet military microbiology facility.
Okay.
According to the historical record, a worker removed a clogged filter from an exhaust system for maintenance.
But the next shift didn't know the filter was missing, right?
Exactly. They turned the machines back on without replacing it.
Wow.
And that single oversight released an aerosolized cloud of highly refined, weaponized anthrax spores directly out of the exhaust pipe, just drifting on the wind into the surrounding neighborhood.
I mean, imagine living in a city where a single missing air filter at a factory down the street causes an invisible cloud of death to drift over your home.
It is the absolute worst-case scenario for inhalational anthrax.
Yeah.
At least 66 people died in the resulting outbreak.
That is awful.
And for years, Soviet authorities engaged in a massive cover-up, claiming the deaths were due to villagers eating contaminated meat.
So they tried to pass it off as gastrointestinal anthrax.
Yes. It wasn't until 1992, after the fall of the Soviet Union, that Boris Yeltsin finally admitted a military facility was the source. It remains the deadliest documented human anthrax outbreak from a single point source.
Which perfectly illustrates why the CDC lists anthrax as a top-tier category, a bioterrorism agent right alongside smallpox and the plague.
Absolutely.
Which unfortunately brings us to the United States in the year 2001 in the Amerithrax letters.
Right. In the chaotic weeks immediately following the September 11 attacks, Letters containing highly refined, finely milled anthrax spores were mailed to several U.S. news media offices and the offices of 2 U.S. senators.
Yeah.
5 people died and 17 were severely sickened.
And it triggered one of the most complex scientific investigations in FBI history. And just to lay out the facts of that investigation impartially based on our sources here, the FBI initially focused heavily on a scientist named Stephen Hatfill. He was later completely exonerated and actually won a substantial settlement from the government. The investigation then pivoted to Dr. Bruce Ivins, who was a government microbiologist working at USMRID, which is the Army's Biodefense Research Institute.
Yeah.
However, Ivins died by suicide in 2008 as investigators were closing in. And because the case was formally closed without a trial, the forensic methods the FBI used to trace the DNA, well, they remain debated among critics today.
And the reason that tracing is so intensely difficult and why critics argue about it is because of how bacteria reproduce. When a lab grows a massive batch of spores from a single parent strain, they're essentially creating trillions of identical clones.
Right.
So finding a slight genetic mutation that points back to one specific flask in one specific laboratory out of thousands of identical flasks across the country is a monumental forensic challenge.
And that forensic deep dive into the DNA of anthrax during the 2001 investigation, it also poured fuel on this massive debate in the scientific community about what Bacillus anthracis actually is. And honestly, this part of the reading completely lost me. I need your help here.
Sure.
Because the genomic sequencing showed that at the level of its main chromosomal DNA, Bacillus anthracis is almost indistinguishable from 2 other common bacteria.
Right.
Bacillus cereus, which gives you mild food poisoning, and Bacillus thuringiensis, which is an insect pathogen used as a commercial crop pesticide.
Yeah.
How can a stomach bug, a caterpillar killer, and a lethal bioweapon technically be the exact same species? How does that even make sense?
It's totally mind-bending, I know. But it comes back to those genetic USB drives the plasmids.
Yeah.
Genomicists argue that because bacteria can swap these plasmids back and forth in a process called horizontal gene transfer, the species boundaries are highly porous.
Okay.
They aren't fixed the way mammals are. So you have a base model of a bacterium. If it picks up the plasmids for the anthrax toxin in the camouflage capsule, it becomes a killer.
Right.
If it picks up different plasmids, it might just give you a stomachache.
So, wait. A harmless food poisoning bug in the soil could theoretically find the USB drive for the anthrax toxin, download it, and become a killer.
It has actually happened.
You're kidding.
No, there have been documented cases, including a severe cluster of pneumonia among metal workers in the U.S., where a strain of the food poisoning bug, Bacillus cereus, somehow picked up the PX01-like virulence plasmids in the environment and caused anthrax-like illness and death.
Oh my gosh.
Because of this, genomicists argue they're really all just one single-parent species with different ecotypes based on whatever genetic cargo they are carrying at the moment.
But I'm guessing doctors and clinical purists hate that idea.
Oh, they push back hard. From a medical and public health standpoint, if a lab technician looks under a microscope and sees the classical markers of B. anthracis, they need to trigger a rapid, unambiguous biodefense response.
Right, they can't be arguing about semantics.
Exactly. They do not have time to debate fluid taxonomy when lives are on the line. They need it classified as a distinct, deadly species.
So if we synthesize everything we've pulled from these sources, Bacillus anthracis really is a masterclass in microbial survival.
It really is.
From Robert Koch's makeshift home lab, where it helped establish the very rules of medical microbiology, to the ingenious armor-plated biology of its spores, to the lethal sophistication of its plasmid weapons, it is an organism perfectly adapted to outlast its environment.
Yeah.
And because humans recognize that incredible stability and chose to weaponize it, it remains an enduring biodefense threat that shapes our national security policies today.
It forces us to constantly innovate our diagnostics, our stockpiling of antitoxins, and our vaccines like the Biothrax vaccine that military and lab personnel rely on today.
But I want to leave you, the listener, with one final kind of chilling thought to mull over. We spend so much of our time worrying about human weaponization, lab leaks, and mailed envelopes. But nature has its own stockpiles, and they are vast. In 2016, there was a severe anomalous heat wave in Siberia, and it melted a patch of permafrost that hadn't thawed in many, many decades. Trapped inside that permafrost was a decades-old reindeer carcass that had died of anthrax long before any of us were born.
And the spores spores easily survived the freezing.
Exactly. The thaw exposed the carcass, those armored spores woke up from their cryogenic sleep, and it sparked a brand new anthrax outbreak in the local reindeer herds and the human population.
Wow.
So as our planet warms and the permafrost continues to thaw across the Arctic, we have to ask how many ancient, dormant outposts of this practically immortal organism are quietly waking up beneath our feet, just waiting for the perfect moment to strike.
A terrifying thought.
It really is. Well, thanks for joining us on this deep dive into the anthrax bacterium. Stay curious, and we'll catch you next time.